In this video, we are going to look at how to quantify non-covalent interactions between biomolecules. It's a super important set of concepts and a super important skill going forward. So let's get into it. So biology loves non-covalent interactions. In this painting here, what we're seeing is all of these different proteins that are all banging into each other. There's there's nucleic acids, there's other small molecules, and they're all interacting transiently. They're running into each other, they're interacting for a while, they're falling apart. And this is really important for cells to do things like respond to their environment, right? They're not just like locked into one configuration. They can change and respond, and the computation that cells are doing is really about a set of interactions forming and breaking and forming and breaking. What that means is we need a conceptual framework to actually understand and study these sorts of interactions. And to make this a little bit clearer, maybe we could just ask two questions. Like if we were to ask, how do we measure the strength of a non-covalent interaction? How would we do it? Like, is this interaction between A and B strong? Is it weak? How do we know? How do we put that on a scale? Another question we might ask would be, I give a cell a drug, or I give a patient a drug in the clinic, and I say, what concentration of drug do I need to see an effect? How would I figure that out? And it turns out these two questions are actually deeply related. So to do this with a real system in mind, we're actually going to use the steroid receptors. I talked about them in my last video, actually. So if you haven't seen that yet, it might be worth going back and looking at it. But if you haven't, or you just want to soldier forward, I'll give a very zippy intro. Steroid hormones are these extremely potent molecules that influence things about animals. So one of the big reasons that a peahen looks like a peahen and a peacock looks like a peacock is actually the relative level of the steroid hormones estradiol and testosterone. And at a oversimplified level, what's going on is, say, in a peahen, estradiol concentrations are high. Estradiol goes into cells and binds to the estrogen receptor. The estrogen receptor then translocates to the nucleus, where it upregulates, say, the creation of genes that make brown feathers. And so that's the linkage between the steroid and the morphology. It's given by which transcriptional programs are activated. But what we're going to focus on today is this interaction here, the interaction between the estrogen receptor and the estradiol. So let's take our questions we pose sort of generically and make them more specific. In this case, we can say, how do we measure the strength of a non-covalent interaction between the estrogen receptor and estradiol? And then what concentration of estradiol do I need to see in effect? So those are the questions we're going to be asking. So let's imagine a cell that has a bunch of different estrogen receptors. So those are the blue molecules floating around. And let's further imagine that we have a huge amount of estradiol. So I'm drawing estradiol in this video as blue circles. And if there's so much estradiol around that every estrogen receptor has its own estradiol bound. We could characterize this using something called the fractional saturation, which is the fraction of receptor molecules that have an estradiol bound. So the concentration of receptor with estradiol over the concentration of receptor with estradiol plus everybody that doesn't have estradiol bound. Now, just a side note, I'm going to be using theta for this. There's a couple of different symbols that occur in the field, but I find theta is the most useful, and I'm going to use it across all of my content. So theta, when I use it, is always going to be fractional saturation. Okay, so here's our molecules. Again, we can see our fractional saturation is 1. Every single estrogen receptor has an estradiol bound. Cool. Now let's imagine we get rid of all of the estradiol. There's some process, the estradiol concentration effectively drops to 0. Now our fractional saturation is going to be 0 because none of our molecules of the estrogen receptor have estradiol bound. We can see that over here on the left here, right? We can just imagine this is a high number now. These are both 0, so we've got 0 over a large number. It ends up being 0. So let's put those onto a graph, both of those extremes. So we've got the high estradiol concentration over here. It's got 1, low concentration is 0. Cool. It's kind of, I hope, pretty intuitive. What happens in the middle and why? Because this is where all the interesting biology happens, right? Most of the time biology is not we have nothing or we have a whole bunch of something. It's sort of squishy things in the middle. That's how biology works. So what happens in the middle? To go after this, we actually need to do a little bit of general chemistry, I guess you could call it. We have to think about an equilibrium constant for this reaction. And so if you don't, if you've yeah, what I'd recommend is why don't you pause for a second, if you can, and think, what is the expression I would write for this equilibrium constant? And actually, I should have mentioned this before I asked you to pause. I'm going to replace this complex with mx, macromolecule plus a small molecule x, macromolecule alone, and x alone. So we can write this in symbols. That's how we're going to write it. So what would the sort of algebraic expression be for the KD? So pause and think about that for a second. Okay, assuming you came out of your pause now, this is going to be mx over mx. It's the products over the reactants. That's the sort of basic definition of the KD, of this equilibrium constant. And I'm going to do a whole sort of block of videos. The next set's actually going to be about thermodynamics and equilibrium constants. So if you're fuzzy on these concepts, don't worry. We're going to sort of unpack those more later. But for the moment, all we need to know is it's products over reactants. Okay, the other thing we need to just mention here is you might have noticed I wrote this reaction as mx going to m plus x, or going to go back a slide, estradiol bound to receptor going to estradiol falling off of the receptor. And this is actually a convention across biochemistry that's a really important common convention, and it is we use dissociation reactions. We always write out these reactions as something falling apart. And why we do that is actually going to become clear in a little bit. But just so you know, that's always going to be true. So when we talk about this equilibrium constant, we are talking about a KD, or the equilibrium constant for dissociation. So KD dissociation. Okay. Feel like we just popped down into the weeds a little bit there. So let's come back out of them. What are we trying to do? We are trying to describe what happens when you go from having, say, a bunch of estradiol around to having very little estradiol around. We're asking what happens in the middle of this curve, mapping fractional saturation to concentration. And we just figured out this KD thing that we're going to load in. So we know this dissociation constant, which is the concentration of M times concentration of X over MX. All right, so what are we going to do? What we're going to do is try to write an expression for fractional saturation in terms of KD. So we're going to use the KD to explain the fractional saturation. So here's our expression for fractional saturation, now written with this MX notation rather than drawing pictures of receptors and estradiols. And we have our expression for KD. Now, I would recommend that you actually try to do this yourself, so that you pause the video and say, I'm going to try to do this operation. And the operation we're trying to do is to write a simple expression where theta is written in terms of KD. So try to figure out some algebra to relate those two. That would be good for your soul. I always think math is good for your soul is how I think about it. But so I'd recommend you try that. So why don't you, again, we're trying to write an expression for theta in terms of KD. Pause. Okay, I am assuming that you unpause. I'm assuming that, and cynically, most of you didn't do it, but that's okay. We're going to do it. We'll do it here. Okay, so what are we going to do? We are going to do a simple rearrangement. We're going to flip KD and MX. So now I've written MX in terms of this blob. I'm going to substitute this blob in up here, right? Because those are equal to MX. What that does is it creates this lovely looking expression down here. I'm going to move forward a slide, just move it to the top. So I didn't change anything. I just moved that expression to the top. And now what I have is something that's got M times X over KD, M times X over KD. What do I see? I see, well, there's a lot of M's in there. So why don't we factor those out? So I've just pulled the M's out to the front. And now I've got M over M and then some other stuff. That cancels, right? Because that goes to 1. And that gives me this expression. We're almost there, but there's some dividing of stuff, and I don't like that, so let's get rid of that. So now I'm just going to take this expression and move it to the next slide. So I didn't change anything. I just moved it. Last step that we're going to do here is multiply this entire thing by KD over KD. So we're multiplying it by 1, basically. And what that's going to do is it's going to cancel this KD, cancel this KD, and it's going to put a KD there, right? Because we're multiplying it across, yielding this, which is our final expression. Okay, so now that's cool, because what we now have here is we can describe fractional saturation, this sort of description of how binding's happening, to an expression with the concentration of our small molecule, and this is our concentration of estradiol, as I've written it. Estradiol, I can't spell. And then this is our equilibrium constant, our KD. Now, I feel like there's lots of asides in this video. I did make a simplifying assumption in this derivation. For those of you that are, say, biochemistry major or care a lot about the quantification of these things, which I think is worth it, you should totally check out in the description. I have a link to the PDF where I actually go through the whole derivation. So check that out if you want to. I felt like going there would be too many asides on asides for this video. So, okay, again, we have this expression that says, here's our fractional saturation as a function of our dissociation constant and our estradiol concentration. Cool. That puts us in a position now to actually start filling out this curve. So we already know this number, it's 0, right? With no estradiol, we have no binding. We already know this number, it's going to be 1. And notice I put a real value in here now because somebody has actually measured, actually multiple people have measured this equilibrium constant. And the equilibrium constant for estradiol binding to the estrogen receptor is 0.1 nanomolar. So 0.1 nanomolar is a known value. And so now we're basically home free, right? We want to know what is the relationship between our estradiol concentration, our X concentration, and our fractional saturation. We have an equation and we have a number. And so we just have to plug some numbers in. So what I'd like you to do is calculate these numbers. What is theta at X equals 1 nanomolar? What is at 0.5 and what is at 0.1 nanomolar. So this is a great time to pause the video and try to fill in this curve yourself. So why don't you pause it? All right, well, we're back from the pause. So here, let's do this. So let's say what is at X equals 1. That's going to be theta equals 1 over 0.1 plus 1, right? So what I just did there was I just plugged in 1 there, plugged in the 1.1, and so that's going to be 1 over 1.1, which is about 0.9. So at 1, we're around 0.9. Let's do X equals 0.5. That equals, oops, that doesn't equal. That is theta equals 0.5 over 0.1 plus 0.5, which is going to equal 5/6, which is about 0.8. So 0.5 is there. And then finally, let's do this one. This one I'm going to write it a little bit bigger. I've got at 0.1, X equals 0.1. That's going to be 0.1 over 0.1 plus 0.1, which equals 0.5. So that's 0.1, 0.5. Okay, cool. So we plugged in some numbers. We got a curve. I'm going to fill it in. A little bit rough because it's hand drawing. But all right, we now know what the shape of our curve is. Remember, we started with this. Oh, look, it's 0 down here. It's 1 up here. What's it look like in the middle? Now we know what it looks like in the middle. It's got this kind of sort of rising, flattening shape. And it turns out that curve shape is really, really important and happens all over biochemistry. In fact, it is the curve in biochemistry. If you're taking a biochemistry exam and you are looking and they're like, draw a graph, and you're like, I have no idea what to draw. Draw this curve. This is like your 85% of the time you'll be right. And the reason you'll be right is because this reflects a fundamental fact of biochemistry. It's saying we are running out of something. Right? When we're over here, we, as we start adding things, we're adding molecules that are binding to the receptor. Once we get out to here, there's no more receptor left. We are out. And it turns out biochemistry or cells are running out of things all the time, right? They make a small number of say, receptors. Those receptors all load up. They make a small number of enzymes. Those enzymes all load up. So we're going to see the saturation curve happening over and over and over in biochemistry. Oh, here, saturation. I had a cool picture of it, right? Once we're here, we are out of receptors to bind to more things. Okay, I don't know if you noticed that there, but we also noticed that the KD is the X that gives half saturation. So recall that I said the KD equals 0.1 nanomolar. That was measured by a group. And what we did is we plugged in X equals 0.1. And let's just do it again to be clear. There's our relationship. Theta equals x over x plus kd, which equals 0.1 over 0.1 plus 0.1, which equals 0.5. Cool. So what that means is that kd is the x that gives half saturation. So right here, x equals 0.1 equals the kd. All right. Why does that matter? What does that tell us? Well, when we ask this question, remember we asked these questions at the beginning. How do we measure the strength of a non-covalent interaction between the estrogen receptor and the estradiol? And what concentration of estradiol do I need to see in effect? The answer to both of these questions, it turns out, is the KD, the dissociation constant. The KD is how much of a molecule X do I need to interact with a molecule M. A low KD means that M has a high, it's backwards, has a high affinity for X. It takes very little X to C binding. A high KD means that M has a low affinity for X. It takes a lot of X to C binding. So the KD is telling us about the affinity of the molecule. Does it want to bind? Does M want to bind to X or not? And so let's actually plot that on this curve with some changes. So this is the curve that I already showed you, low KD with the estrogen KD. So let's imagine now we have a molecule with a lower KD. What will that curve look like? Where should I draw that? So the KD has gone down. What does that curve look like on here? So maybe if it's not obvious, pause and think about it. Where am I going to draw a curve with a low KD? All right, I'm assuming you unpause. The answer is this. It's going to be even steeper, right? Because we have a lower KD. It's going to take even less estradiol to bind, and so it's going to be shifted to the left. How about a high KD with low affinity? That one's going to shift this way, right? The KD for both of these is the concentration at which we are at 50% saturation or half of fractional saturation. And so this is our, as I've sketched it here, our high KD, our low KD is down here. And this is, our real KD. Okay, so these curves are measuring affinity by basically what concentration of estradiol does it take to bind to the receptor. Another interesting thing about this is that the KD of a real protein often corresponds to the physiological range of whatever it binds to. And so this is the real KD. This is the binding curve for the estrogen receptor. Turns out this is the reference range. If you go and say Mayo Clinic, which is a really cool website, the Mayo Clinic website has reference ranges for basically any molecule you can get a blood test for. This is the reference range for estradiol in a human adult female. And let's look at this. What does this bracket? Well, it basically brackets about theta equals 0.3 to whatever that is, about theta equals 0.9. And so when estradiol concentrations are low, the estrogen receptors are not bound, or only 30% of them are bound. And then when estradiol concentrations are high, 90% of the estrogen receptors are bound. What this means is the cell can respond, right? It can detect a change in X. Imagine if the concentration range of the estradiol in the blood was out here. It could fluctuate around all it wanted, and the cell could never tell. It would always be loaded up with the estradiol, right? But because this range sort of brackets the KD, because it's right around that equilibrium constant, a cell can know, oh, estradiol concentrations are low because I've only got 30% of my receptors bound. And then up here, it's like estradiol concentrations are now high because 90% of the receptors are bound. Okay, I want to make one last point on this, and that is that KD can also be used to quantify a receptor's preference for different molecules. From my last video, in my last video, I talked about how the estrogen receptor can tell the difference between estradiol, this steroid we've been looking at, and testosterone. And just to show schematically here, this is a estradiol bound in a nice way to the binding pocket. Testosterone does not bind as well. It loses a set of hydrogen bonds at the A ring of the steroid. So if we go back and forth, hopefully what you can appreciate is the testosterone binds worse than the estradiol, at least looks like it's binding worse. And it turns out we can see this in terms of the KD. So this is reflected in the KD. So these are the measured KDs of the estrogen receptor for estradiol, 0.1 nanomolar. And for testosterone, it's 100,000 nanomolar. It's way, 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 way worse. And in fact, if you look at the binding curve here, at these concentrations, up to 4 nanomolar of, I guess this shouldn't be this should just be steroid now. At 4 nanomolar estradiol, it's fully loaded. At 4 nanomolar testosterone, it hasn't bound at all. Nothing has bound because the KD is so high. So the difference in the KD for these two things tells you that it has a high affinity for estradiol. It has a very low affinity for testosterone. Okay, so we can measure the strength of a non-covalent interaction with the KD, the dissociation constant. And this tells us the concentration at which half of the receptors are bound. So the low KD implies high affinity. The high KD implies low affinity. So it feels a little bit backwards, but it's because that KD is telling you the concentration of half binding. We can measure, and finally, we can actually think about this. We've talked about knowing the KD and then using it to measure things, but you can actually think about it the reverse. This is where people get KDs. You measure fractional saturation as a function of something, and then you find the concentration that gives you half saturation, and that corresponds to the KD in this curve. All right, so that's what I've got. Hopefully you've got a better picture now of how to think about and sort of measuring and quantifying non-covalent interactions, some beginning of a notion of how to work with KD. We're going to do a lot of it throughout these videos. So I look forward to doing that with you and hope you have a wonderful morning. Yeah, you're definitely a morning person watching this.